My name is Ray Browning. I am grateful to be part of a small team of people at a company called Biomodem. My co-founder is Zach Lerner, and I'll start by giving him really all of the credit for the technology that we're working on. Zach was a brilliant member of, I used to be in academia in our lab, and he's gone on, I think, to really try to um, capitalize on the advances in robotics and the advances in electrification of transport. I'll use those two things as two factors that are allowing us to build what I would call active assistive devices for individuals with neurologic conditions. In particular, we're focused on providing assistance, powered assistance at the ankle joint, and we have been focused on uh, uh, children, adolescents with cerebral palsy to improve their walking function. So we've been at this for a few years now, and have good evidence of effectiveness in a rehabilitation setting.
Yeah.
But obviously, we're excited about the possibility, and this is where the Blueprint MedTech support comes in extending the reach of that technology into a device that goes home with a family and has really two functions, which I think is important. One, as a neurologic rehabilitation function, so to continue. Oftentimes, when people are done with their rehab, that's it. They don't really, and they're given assistive technology that supports their current level of function, but isn't necessarily intended to improve their function over time. So we think these kinds of systems that we're working on can actually do two things, can provide ongoing and continual rehab so they continue to improve their function over time, but also to provide daily assistance in activities of daily living that these individuals encounter. In particular, things that are really challenging, like stairs or ramps or
Yeah.
uneven surfaces. So, and in a younger population who wants to be mobile, that's really, really important.
Sure. Yeah, no, I really want to dive into that. But just to keep it kind of maybe sequential, I wanted to step back for a minute and maybe zoom out all the way to the beginning. If you don't mind sharing, how did this genesis of this idea come together? How did the founding team meet and start this mission together? If you could speak to that, that would be
Yeah,
great.
I can, Santosh, and I think I'll start by saying that both Zach and I, as the two co-founders, have a long um, belief in the power of human movement to unlock potential. And that sounds a little, trivial, but uh, we both we came together with that sort of shared belief that, wow, uh, movement has a really profound effect on how people see themselves, how they see the world. And as we have thought about that, the mission has gotten even deeper where um, the when you go to see someone, a friend or a family member, or you come home from the end of the day, oftentimes the last thing you do before the hug or the handshake or the hello is you walk. And so mobility is a form of human connection. It's a way that we reach people to connect with them. And our mission has really kind of broadened from, hey, we make devices that improve, neurologic function in these individuals to one that empowers these individuals to have these human connections. And we know that individuals with disability are often more isolated. They don't have the social connections. And that's really, really important to us. And so Zach and I took this broad interest in movement, and we were working a lot with children. We had a number of studies which we both really enjoyed. I mean, it's so fun to work with young people. And Zach was really, really interested in, he was designing artificial muscles, in the lab, kind of on his own, just made this horrible pneumatic air compressor based muscle thing that didn't, it was a really creative idea, but you had to lug around like a 45 pounds of, hardware. Um, and,
Yeah.
he we parted ways. I went off to work at Nike in innovation from academia, and Zach went to the National Institutes of Health to do his postdoc.
Yeah.
And about 5 years ago, we reconnected, and he said, "Hey, I've been working on some really interesting stuff. Um, I would like it to see and reach a broader audience. I would love to see it commercialized. Would you be interested?" And our in our laboratory at the university, because I've not always been an academic. I've had a lot of other life experiences. We were always doing industry sponsored work. And so the students became really familiar with the fact that there was NIH, NSF, there was the funded, federal work, but there was these other organizations that were trying to make products that met the
Yeah.
needs of their end users. And I
Yeah.
think that experience gave Zach the window into saying, "Oh, wait, you actually can take the knowledge and then translate that knowledge into something that might be useful."
Yeah.
And then you can extend, if it is useful, into something that might be commercially viable for people. So I really am grateful to him for kind of reconnecting and reaching back out. And that was about 5 years ago. And then we've been working on this project ever since together.
That's great. And, a lovely story, so to speak, of you enjoying the work with adolescents and um, teenagers or, even younger
Mhm.
populations. That's really fantastic. I would say you're one of the unique portfolio companies in Blueprint that's addressing that kind of patient population. And so maybe we can dive now into a little bit of the solution and how you arrived there. So I certainly appreciate this is going to be applicable to broader age groups. But any reason why you decided to target and or maybe start in the younger population?
Yeah, there were a couple of reasons. It's a really good question. And those reasons were um, we were convinced that the lifetime benefit of even making incremental improvements in mobility at a young age for this population was really going to be life-changing. I mean, the trajectory, a small change in mobility function translates to employment opportunities, educational opportunities, relational opportunities. There are so many downstream benefits, and we thought, gosh, if we can help these young individuals who and the evidence just wasn't great that these kids were, many of the individuals with moderate cerebral palsy and certainly more severe cerebral palsy end up in a wheelchair as
Yeah,
wheelchair users.
right.
And that isn't a bad thing necessarily, but it's not a desired outcome. It's not something that they want. And so there was that. There was the younger age has a longer beneficial,
Yeah,
trajectory.
yeah.
And so that was one, and the other was we ended up talking to a lot of these caregiver, provider, and patient triads, and nobody was very satisfied. They were like, can't there because there's something else we can be doing. And that, as you know, working in innovation, that's something you're really listening for. Is there an unmet need? And
Yes.
one of the differences, I think, Santosh, in a younger population is you're not dealing with, just an individual. You're dealing with a care team, and oftentimes that's parents, siblings, obviously the provider teams, and um, they have in younger ages, there's a sense of urgency to solve the problem,
Hmm.
which you don't see as much in, older populations. For example,
Yes.
maybe survivors of stroke. They
Sure.
want to get better, but there's a different sense of
Yeah.
now. I want,
Right.
can we bring this to market, or can we have
Yeah.
access to this now? So
Right.
those two things, one, the impact, the potential lifelong impact, combined with the sense of unmet need and urgency, I think were two of the things that really have kept us. And I mean, I will say, uh, it's not a recommended pathway. Pediatric medical device is super hard. I mean,
Right.
because
Sure.
you've got, regulatory
Right.
challenges and other
Yes.
things. So, um, but
But
I
that
do.
aside, this is pretty fantastic in terms of impact, as you say. I really appreciate that comment, and I want to just highlight that because you're right. Having the kind of longitudinal impact in someone's life is life changing, as you just pointed out. So there's a lot to be said there, and
Yeah.
it's beyond just the sort of the immediate medical condition. It touches on every aspect of their life, as you just said. So socioeconomic benefits, family benefits, goes
Yep.
on and on. So
Yep.
that's really a key insight that I'm glad you shared with us, that I wasn't necessarily thinking through that perspective and the immediacy. Wow. Absolutely. I can
Mhm.
sense that. If me, for instance, and I think many parents, right, if there's a way to help your child, yeah, you have a very motivated
I'll tell
buyer, so
you,
to speak.
Santosh, I was woefully unprepared for the emotional responses from caregivers, parents in particular. I thought, cool, we're making these wearable robots. The kids are going to love it, and they're going to feel like,
Yep.
Iron Man and blah blah blah. And then do a demo, or maybe it's a clinical trial, and and you look over and the parent is literally in tears,
Emotional. Yeah. I
Yeah.
was I was literally about to say I would not be surprised if I saw tears. I
Oh,
would,
and I was at first I
yeah,
was like, uh-oh, did we do like it's is and they were
something
just
wrong.
like,
Yeah.
yeah, and they would look at you, and we've had many parents who've turned and said, if that's possible, you've just changed my life. What I thought was possible for my child is now different than what it was 5 minutes ago. And I mean, gosh, that's a
That's
uh,
profound. Yeah.
wow. I mean, yeah, it really took me by surprise. I
That's
hadn't
awesome.
I hadn't really
Great.
appreciated
Yeah.
the
Yeah.
and then you sort of become, all right, well, I gotta take care of this idea, right? I mean, we've got to nurture this along. And because
Yeah.
there's times, I mean, you guys know that it's hard, right? Like, how are we
Yes.
gonna make this work? Or that didn't work, or we didn't get that funding, or this or that happens. And there's
Yeah.
plenty of places where you can say, I don't want to do this, but you think back on that responsibility. Whew,
Absolutely.
yeah,
Yeah, that's great. What a motivation for you and the rest of the team. So that's fantastic, and thanks for sharing that.
sure.
So as we kind of get into maybe the solution a little bit, just describe, because you mentioned you have a co-founder who's very technical, kind of came up with a lot of maybe interesting ideas, but ease of use was lacking. How did you guys arrive at the current solution you have? What zoned in? What brought you guys to zone in and see this as the solution to tackle?
So we, you know, um, we're really experts in lower extremity neuromuscular mechanics. And one of the things that you learn when you think about, the most common form of using our legs to get around is walking. And
Hmm.
when you really understand walking, you pretty quickly realize that, you use this kind of inverted pendulum of walking, right? We have a stiff leg, you push with the trailing leg, your center of mass goes up and then it comes back down again, and then another leg is out in front of it. And as a result, the ankle is the primary pusher, right? It's what pushes the center mass up and over. So it's a really, really important joint. And the strategy oftentimes with these kids is to lock that joint, is to actually put it in an ankle foot orthoses and lock it. And that provides a supporting function, but it eliminates any ability to use the lower extremity to actually 40% of our power output in walking comes from the ankle joint. So it's a pretty significant loss. So it wasn't hard to say, if we can target that joint and provide some active assistance in that joint. The
Hmm.
challenge that you run into is that current systems for actuation are heavy. So motors are heavy, batteries are heavy, and muscles are incredibly strong. And so if you're augmenting or replacing some function of a complex like the calf musculature, you need powerful motors because those muscles provide amazing torque around the ankle joint. And so a lot of our effort has been, how do you package the mass, batteries, motors, and all the other sensing
Yeah.
equipment in a system that isn't too heavy, because if you have a 5 kilo device on your legs, it's not going to
Yeah.
work.
Right. Right. So you have sort of
Um,
competing goals here. You want
yes.
a lot of force, but you want extremely low weight.
Yep.
Sort of specifications that you're coming in with are um,
Yeah.
pretty
And that's
interesting.
been that's been an interesting evolution. When we first started working on this, it was the strategy was let's put all of the horsepower, no pun intended, batteries, motors, control boards in the waist, and then run cables down to the leg assembly and actuate the leg assembly with
Mm,
Bowden
yeah.
cables. Great idea, works well. You end up with a light lower extremity assembly, except that when you put it on a table and you ask people to, try it, they won't. And they won't because
Yeah.
it looks complicated. It's long. It's like, what are these cables? Oh my God, they're everywhere, and so this is one of those that the technical solution wasn't the usable solution. And we fought that for quite a while, as engineers always do, right? Well, but it's the best solution.
Yeah.
And then my job has increasingly been voice of customer because I spend a lot of time talking to people.
Yeah.
And I was like, yeah, I agree, but if people don't pick it up to use it, it doesn't matter how well it works.
Right.
They have to get it on. So we've
Yeah.
subsequently been working with Bluetooth MedTech support to bring that actuation system down below the knee, but keep it more proximal. And that seems to be a fairly viable solution. But I wish we would have made that transition a little bit sooner. We waited a little bit longer than we should have. But so
Yeah.
essentially, what we're making, I think the easiest layperson's description is we're making an e-bike for your feet.
Nice.
So,
Yeah. I like that.
you push on it like an e-bike, you get
Yep.
assistance, right? As your foot comes off the ground, you get
Yep.
dorsiflexion assistance, and then, rinse, repeat. And the controller it's a proportional closed loop controller. So
Yep.
it responds to how you move. It's not prescriptive. So it's not doing the same thing every
Yeah.
step. It's responding to how you're
That's really
moving, which
nice. So it's a nice
we've
closed feedback loop there.
yeah,
Yeah.
we use two sensing elements, one to kind of estimate the demand and the other to measure, and then we're closing that loop. And by using a sensing array in the foot, we're able to dic to address changes in locomotor desire. So if it's a
Yeah.
staircase or a ramp or changes in speed, the amount of assistance that's provided is changes depending on those tasks. And I really think Zach has done a nice job in building an adaptive control system that isn't trying to say, well, wait, is he about to walk? Is he walking? Is he going up and down stairs? Because classifiers are really tricky, and they're much more tricky in children than they are in any other
Yeah.
group.
Yeah.
So, our approach is to provide the assistance for the way that the individual is moving, not prescribing the way that they should be moving.
Right. No,
Yeah.
that's a great approach. I like that. You guys have really landed. I'm sort of kind of figuring out the best solution for this group of patients. I mean, I personally remember seeing the device, so I picked it up, and I remember being kind of blown away by the mechanical capabilities, but yet at that weight sort of the footprint of the weight
Hmm.
was a lot lighter than I expected. So you guys have
Yeah.
done a great job of miniaturization and bringing down the weight of the overall product as well.
Yeah, we're
So
working on it, and so thank you. I mean, I feel like um, we're going to continue to see advances in actuation systems. That's going to happen. And so what we will be able to capitalize on is those we can integrate those advances.
yeah,
It's not so much having to start over again. It's like, oh, cool, there's a lighter motor with a lower power, with
yep.
adequate power, a battery density is going to continue to go up, which
Continue to get
means battery
better.
size
Yep,
is going to go down.
yep,
You're going to get more efficient controllers. And one of the things I think that's really exciting about what we're working on, and this really sort of fills the entire kind of electromechanical assistive space, whether it's upper extremity stroke rehab or is, we have a control approach which is really based on, minimizing metabolic cost of walking. That is one approach. Another approach could be maximizing stability. Another approach could be, enhancing range of motion. And what's really exciting is once you have a tool like this, you can modify the parameters by which you are
mhm,
optimizing. And I
yeah,
think it's something I try to articulate to people is this is just the first time, the first pass. I can envision devices in the future having um, multiple control algorithms at the disposal of the microprocessor to account for, oh, this feels like an unstable environment. Let's switch into stability mode so that the person has a lower likelihood of slipping or falling.
Oh,
Um, or this is a Parkinson's patient who needs a very different sort of controller. They're not really
yeah.
interested in minimizing metabolic costs. They're interested in not freezing, right? So
Yes.
that's the other thing is when you get these electromechanical systems on the body, oh my gosh, I mean, there's so much we're going to
Yeah,
learn.
yeah, that's exciting. Yeah, no, it's that's really neat. And you already brought this up, but I think your background is quite fascinating and unique. I mean, having seen the commercial side through the lens of, I can only assume, of being at a company like Nike, and then bringing a lot of that, as you said, kind of voice of the customer and listening to what is really out there, what is needed. I think has done just a world of difference for this team. And so I think you and Zach have a really good kind of back and forth between the two of you. And that alchemy seems like it's really resulted in something awesome
Thank
by
you.
working
Yeah,
together.
it's been yeah, I think as co-founders we have landed on kind of the right combination of roles and responsibilities, as we move
Yeah,
forward. And
absolutely.
I've really, I do have to remind the team every now and then that I do know a little bit about musculoskeletal mechanics. It's kind of funny, but I'm busy running the actual business, of
Yeah.
it. So,
They- They've got
yeah,
you labeled as the shoe guy.
yeah, exactly. Or I'm the crash test dummy. Like, can you walk in this now? We want to. I'm like, yeah, I'm happy to walk in it,
Yeah,
but I can also provide you some feedback on whether
do
that's
other
going to work or
things.
not.
That's
So, but
great.
uh,
Yeah,
that's okay.
good.
That's totally fine.
Yeah. So let me turn again back to kind of the company, and you've been with the Blueprint hubs now for a couple of years. Can you just speak to how the experience has gone with working with Blueprint and what you think you've gotten out of the program, and just comment on your thoughts?
Oh man, I get to tell the story quite a bit because when we talk to business development or even strategic partners in our space, in the bracing space, or investors, um, there isn't a lot of awareness necessarily of Blueprint MedTech. And when I say, remember that we developed a vaccine portfolio in an absolutely remarkably rapid period of time, at the turn of this last decade, and unprecedented. It was just
Yeah.
like,
So
and I
Radix,
said,
absolutely.
so
Yeah.
yeah, so Blueprint MedTech is the
Next iteration of Radix. Yeah.
exactly, and so I say, so it is. People don't know that and they're like, oh my gosh, and so we're trying to accelerate these advances. I mean, from a uh I don't know that we'd be here without it, quite frankly. The support has been tremendous, and the thing that's nice about the program is it's a combination of the typical direct support. So we're making things and we're using the resources that the program provides, which is fabulous. But we also have the in-kind support, which has been really, really important. So NIH has agreed that there are other vendors that we can use for certain parts of the program. And that has been absolutely critical. And those, even right now, we're engaged in a pathway to payment conversation with an experienced person in reimbursement strategy for this device. So. Um, that's been, and then we have the mentorship. So we have Tony who's on our mentorship team, and he has been guiding us through the project. And then, another thing that I think has been really helpful for us is we can say to strategic partners and investors that we're a part of this program, and that does two things. One, it de-risks the opportunity because the program is taking on some of the capital requirements that are necessary to build this kind of technology. And we're also getting access to the expertise and regulatory reimbursement and all the other pieces. But I think almost more importantly is the validation that this is a program or a project. What we're working on has been validated by some of the brightest minds in the neurotech space, and we are a part of that group. So what we're working on, and that is a very powerful message to the community because they're like, okay, got it. Yeah, I mean, this is a really this that has helped us significantly in our efforts.
That's fantastic. I'm glad to hear that. And we're excited about continuing to help further grow and expand your project and hopefully get you, even further, with our partner program of the
Yep.
translator. I'm sure you've heard about that. You'll be
Oh,
able to
yes,
do
we
that,
have.
I'm sure, soon. Yeah, I'm sure you're
Yeah.
looking at that
Yep.
and certainly take advantage. But as you look out a few years from now, and it sounds like you may be already doing it, you're thinking about bringing more money in. What does the future of the overall company and the trajectory look like? What do you see as you look out 4, 5, 10 years?
Yeah, for the next 4 to 5 years, the focus is really on we have I won't go into a lot of details, but we have not just the active ankle assist device that we're working on, but we have some passive devices that I think are really, really interesting. Um, and we've started to assess other joints, as possible sites for intervention.
Yeah,
But the real focus is right now is in that validation, finishing, making products that can get through the various hurdles of product development
yeah,
into the hands of providers and patients, both in a rehabilitation setting as well as, and the Blueprint MedTech support is for the personal use or the home use of a device. So a system that becomes prescribed and fit by an orthotist and then is used by the patient for both rehab and assistance. And so our focus will be on building the product, getting the clinical validation that this home environment
yeah.
is works, and then building the necessary infrastructure to deliver that into paying customers. So how
Yeah.
does that look? And that's where we're already talking to the big strategics in the orthoses and prostheses space to say,
Yeah.
hey guys, this is coming,
Yeah.
and are you ready to take it on? And we're getting some really positive. We've had some really positive conversations with players in that space, large entities that have distribution networks and sales and fabrication, and they understand marketing and reimbursement. And our perspective is we would rather partner with a group like that and take advantage of those things that they offer, as opposed to trying to build that ourselves, right?
Sure.
So I think there's partnership is really, really going to be important for us for commercialization success.
Got it. Excellent.
Yep.
Yeah, no, you're right. I understand that space. Not been in it personally for some time, but I remember looking at the prosthetics area at another sort of period in my career, and it is amazing that the amount of value that you can create there, but it's just a different industry, sort of structurally, may not fit into the mold, as you point out, as a traditional kind of, hey, we're going to raise a lot of money, build this large
Right.
company, and then it's an M&A exit or something like
Yep.
that.
Yeah.
But nonetheless, there's innovation needed for these patients. Although these patient populations, as you know, are small, they're not your typical billion-dollar maybe market valuations, but nonetheless, very underserved
Yep.
populations
Yeah,
that
that's right. And, and that's a double-edged sword, of course,
yeah,
because you when you're going out raising the money that's required to bring something like this to market, you do run into that skepticism of, is it enough people? Can the company be large enough? Is the market size big enough?
right.
And one of the things that I will and I'm bringing this up to strategic partners, and they're not quite ready, to hear this. But my point has been, uh, if your portfolio in 5 years doesn't include a majority of smart devices, meaning smart bracing,
Mhm.
uh, you're not going to get reimbursement
Right. Yeah,
because CMS and Medicare and the major payers are going to go, well, wait, you're making this brace and you don't even know whether it's being worn.
Yeah.
Why are we going to reimburse for something that we don't even know if it's actually being used? And that is kind of the current state, for example, of bracing or even prostheses. The companies can't tell you whether the devices are actually in use and how they're used. And I'm like, look, that is going to change.
Yeah.
And if you think that a thermoplastic brace that you throw on a kid is going to be enough, I'm like, I want to convince you that lead it. Don't be
Yeah.
forced into adopting it. Do
Yeah.
it now so that you have a portfolio of these smart products that will teach us all how do we make things that people will use and benefit from.
Yeah.
Um,
Yeah.
and orthosis and prostheses is a little behind in that space. Prostheses more so. I mean, you're seeing more advances in prostheses, upper extremity prostheses in particular, are really getting
Right.
fabulous. And then you've got powered ankles, powered knees. So that's going to happen. And it's fun to actually, kind of challenge these strategics and say, wouldn't you rather have a smart device that you could fine tune and know and really and be able to show payers like this is why it's working? So that's coming.
That's great. Yeah. And I think you're absolutely right. That's going to definitely be the sort of future of combination of things, personalized medicine on some
Hmm.
fronts.
Yep.
And also, you said that down to like all these diseases that present in various ways, but mobility is a certain aspect or condition that many diseases share. And so I think there's a huge demand out there that is maybe latent just because the right
Yeah,
solutions are not there yet.
I
Um,
agree.
yeah,
And people are asking for they're asking for better solutions, and they're doing it in some ways directly, but in other ways indirectly, and they're doing it in a expressing a disaffection with the current
yeah,
solutions, and that's another way of saying, well, if there was a better solution, I would
right,
migrate to it.
right, right.
And you're right. I mean, just what is it? 70% or 50% of all individuals over the age of 75 uh, struggle with walking.
Yeah,
That's a
exactly.
lot of people.
A lot. Yeah. I mean, that's a huge Medicare population also
Yes.
overlap. So
Yeah.
lots of interesting things going on there. So I think and from an engineering standpoint, your solutions are going to only get stronger, lighter, and
Yep.
so even more addressable market to your point
Yep.
about like these kind of populations. So yeah, that's fantastic and it's exciting future that I see for you all, and it's really exciting that we have some time together in our
Agreed.
Creator Hub to help you along the way to help kind of bring some resources and usher this along. This in many ways, I think, is a exactly the kind of prototypical idea that we had when we started, as you pointed out, RadX, getting into Blueprint. What we wanted to accelerate in terms of innovations to market. So.
Yeah,
Uh,
Santosh, and I think in this day and age, in particular, people need to understand that there are really, really amazing things that taxpayer dollars can help support. And they are, and they have been for a long time. And I think in this era of a little bit of just, questioning that, the value of that investment, um, I would like people to know that we are immensely grateful to the taxpayer.
Yeah, that's great.
Right.
I
You know,
really
that's a
appreciate that
this
sentiment.
is that has what has made this country so innovative is that we have a shared acceptance of, yeah, some of what I make goes to here, but it's going to this program, which is building these things. And
Yeah.
I just feel like we really need more of that message to get out to people,
Mhm.
that it's not wasteful. It's incredibly, and you have to make bets, and some of them work and some of them don't,
Yeah.
but all of them move the ball down the field. I
Yeah.
mean,
Yeah.
so
Yeah.
you know,
Absolutely.
yeah.
No,
And the
absolutely.
institutions like Johns Hopkins and Harvard and other institutions are uniquely positioned to support these kinds of initiatives. I think it's not fair to the scrutiny. It's like I there aren't very many places that can pull off the support that's required for these kinds of programs. So.
No, thanks. I really appreciate that feedback and you saying the kind words. So we equally are excited to be one of the incubator hubs. We hope the program renews. That's a big
Yep.
part of this next coming year, 2 years. We
Hmm.
want to continue this great work. We've learned a lot by the first few years process of working with folks like you. So even more further convinced there's a huge need for Blueprint. And we are different than other programs, like you point
Yep.
out, at
Yep.
NIH, not like the SBIR program or other programs, which are fantastic in their own right. But what we're doing is just a little bit different. It's
Yep.
focused on resources and mentorship that just we don't see at any other program
Yep.
across the
Totally
board
agree.
at
Yeah.
NIH. So,
Yeah. It's
so
very it's very strategic and a smart way to do it. And it's I can speak from when I tell people about the program, entrepreneurs, in the med tech space, they're really like, what? Tell me more about this. How do I find out about this? How did you apply? Blah, blah, blah. So
yeah,
there's really there are I think as these I would love to see these programs expand. I think we all would.
yeah,
Um, uh, and especially because, when I say, you know, yes, we're doing some earlier stage R&D, but we're also getting the support to build the pathway to market entry and commercialization, and we're understanding reimbursement. We're doing all of these other things. And like, what?
yeah,
In the context of that isn't that typically isn't even allowed in even a phase two SBAR. Like, we can't be
right.
commercialization activity. You got to sort of end here. So super grateful.
Welcome, Christina. Really nice to meet you. So I'm looking forward to our conversation and learning more about your project. I know you're a Pathways candidate and successfully completed the program with us recently. Although we haven't met, I'm really looking forward to today's conversation, learning more about you. So let's get started with if you can just explain how this project came to be, what was the motivation by you and others potentially on your team to start this project, and where you are today with it.
Yes, thank you, Santosh. Lovely to meet you also. I have worked on Alzheimer's disease my whole scientific career, so I have a PhD in pharmacology and neuropharmacology in particular. And what has always been disturbing, I guess, is one word, is that we have known a lot about the disease, but similar information for over 120 years now. So the disease was discovered, if we want to call it that, by Alois Alzheimer when he had a patient called Augusta D present to him in clinic. And the story behind that that really captures my heart is that she sat in a chair in front of him and said, "I've lost myself." And we're very lucky because Alois Alzheimer was not just a physician, but he was also a scientist. And he didn't want to just dispel or discount her symptoms as something that came from magical forces or whatever they thought 120 years ago. We really had no understanding of the brain at that time. And her family were of little means. And so they came to an agreement. Her husband and Dr. Alzheimer came to an agreement that he would cover her care if he could have access to her medical records and her brain post her passing so that he could learn a little bit more about what was going on in her brain.
Yeah,
And that's how we learned about amyloid plaques and tangles. But whilst our field is really trying to dig into learning more about the disease and what starts it, what causes it to progress, we really are still focused on those amyloid plaques and tangles. Um, what's also interesting is the disease can start up to 20 years before symptoms are, visual or physical symptoms of loss of memory or memory lapses start to show up. The disease can have been ongoing for almost 20
yeah,
years. So we need to
absolutely.
pick it up earlier.
Absolutely. Yeah, that would be wonderful. I mean, as a personal aside, we have a family history, and I'm sure many families can speak to this. So, we've observed it. I'm actually going through it as a caregiver slash son, my mother, has come down with symptoms, over the past 10 years. So I'm very well aware of the disease at a personal level. And so are many families. So it's great work, certainly, that you're doing. I think it's an extremely pressing problem in today's sort of, at large population, both US and worldwide. So applaud you on that effort. And so, if we can maybe transition now to learning a little more about what your group's idea is, what is the innovation that you all have come up with and feel that you could make a difference in this field with?
Yeah, so and I'm sorry for your mom. And for you, I think that's the hard part, right? It's such a burden on caregivers because unfortunately the person essentially loses themselves in the disease, but you become very aware that your mom is not your mom anymore. So this really was part of the motivation, not only for us as individuals, but for people who are concerned about developing Alzheimer's. We wanted to develop a blood test that would allow for monitoring, preferentially monitoring over your lifetime, so that you would know when your brain is showing alterations that may indicate disease. And that is scary to some people when we don't have therapies, but it's not really true that we don't have therapies anymore. We have two anti-amyloid therapies that are being produced by Lilly and Eisai. Um, and if those were given earlier, they may have more beneficial outcomes. So at the moment, we're focused on them trying to reverse amyloid that's in the brain and seeing if that can then improve, the physiological health of the brain. But if we can pick up earlier and give those earlier, that would be great. But what is prohibitive to that is that our current testing methods are PET imaging and lumbar punctures, which, when you think about cost and the requirement for physicians to be sure of what they're asking an insurance company to cover, they need to, you need to have very clear symptoms and know what
Yes.
you're working with the patient to be able to diagnose or
Yes.
say that you need to do a test that's it. Whereas the blood test, you can do a blood test
Yeah.
annually. If a
Yeah,
family.
these tests are too far along for the current clinical workflow, as you say, which is clinical manifestations have started to warrant this kind of expensive testing. As you clearly pointed out, the disease progression has been going on for potentially decades, and so huge unmet need in terms of early diagnostics. So it sounds like that's really was the motivation for you. And
Absolutely.
so, in terms of the diagnostic development, can you speak a little bit about that and what your group has developed?
Yeah, so it's a plasma-based test or a serum-based test, so a blood-based test. But
Yeah.
what we're taking out of those bloods are little lipidy, bilayer lipidy vesicles called exosomes. And these are advantageous. So we're aware that we have an FDA-approved p-tau 217 test that has, I think in March this year or April this year, been approved. The problem with a single analyte test is that it's measuring one thing. And what exosomes can do is they carry cargo. So the cargo reflects the cell from which it was produced. And so instead of just taking all of the exosomes that are present in plasma, which are 10 to the 11 particles per mil, it's a trillion particles in every mil of your blood, we wanted to isolate the exosomes that came from the brain. So I designed and tested the ability to just pull out exosomes that came from neurons or nerve cells and those that came from the inflammatory cells, so microglia. So we are
Hmm.
becoming aware of the role of inflammation, obviously,
Right.
in Alzheimer's and trying to address that. So it's really looking at the health of these brain cells, but from the blood.
Absolutely. Great. And when you started this research and development effort, to what degree have you narrowed in on the exact biomarkers? Is that still in development? Can you speak to that a little bit? And I'm sure there's other groups, not just yours, but if you could just share kind of what's the leading prevailing theories of what we should be looking for at this early diagnostic phase.
Yes, so we have filed this as a full patent. So I think in January is when the lawyers decide what prior art exists out there. But what we did is we covered the process too. So I think what was different is people and people are still doing this, and it's still important, are isolating exosomes and working out what they are carrying, what pathology do they have on them.
Hmm.
But nobody kind of took the step back to ask, what does a neuronal exosome carry, though? What does a microglial exosome carry? So, really, it was just isolating all exosomes, trying to enrich them with markers, but not testing necessarily that those markers were actually on an exosome that came from a nerve cell. So, I really just took a step back and said, I'm going to isolate exosomes from nerve cells. I'm going to isolate exosomes from microglia, and I'm going to ask them what they're carrying. And then I'm going to use those markers to pull them out of plasma.
Yep.
And this is becoming something that a lot of people are interested in. So now we're developing a portfolio on autoimmune diseases.
Sure.
We have a portfolio on cancer as well. So
Excellent. Excellent. Yeah, that makes sense. So, in terms of data sets or samples you've acquired, is this something that is publicly available? Are these data sets you're curating in your lab or your partners, collaborators? Can you maybe shed some light on your development to the degree that's, not confidential?
yeah, so we know that so if we think of if we look at sensitivity and specificity and you draw those nice rock curves, a perfect area under the curve is 1.
Mhm.
And even with 10 control subjects, 11 MCI, and 19 Alzheimer's subjects, our ability to detect Alzheimer's people from people who are control or healthy controls, that's a whole other topic, um, is 0.95, 0.94.
Wow.
And then we can tell who has Alzheimer's versus mild cognitive impairment.
Wow.
What I spent most of yesterday doing is increasing that number now to 50. So I want 200 control MCI and AD just for full power.
Yeah.
But everyone that has looked at the data are already excited about what it's able to do. The population that's really more complex are people with mild cognitive impairment because you get amyloid as the first marker of Alzheimer's, but amyloid is present in a lot of neurodegenerative diseases. So that population can be quite variable because they can go on to develop a different type of dementia, because
Right.
obviously Alzheimer's is just one type and
Right.
dementia would be the umbrella. And so we need,
Frontotemporal dementia. Yeah.
yeah, so we need to increase especially the numbers in the mild cognitive impairment. So yes, I spent all day yesterday making more, and hopefully in the next 4 or 5 weeks, we will add at least another 50 to each. So we'll
That's
become
great.
more confident in our data then and seeing what's happening.
Yeah, that's great. Yeah, I mean, exactly. I think all the challenges of the field, you just mentioned, being able to parse out other neurodegenerative diseases that are mimicking, from a biomarker standpoint, very, very similar to what you see in Alzheimer's related dementia. So that's great. And going back to our clinical conversation, MCI absolutely, I think, is going to be so important as we try to move this diagnosis further upstream, if you will, and in terms of earlier identification of patients. So I applaud your work. I hope it does continue to bear fruit in terms of the earlier the MCI can be detected, or if even better, if some of these biomarkers could be detected even pre-MCI, if we could use it as a general screening tool. Is that something that you hope to eventually take this innovation towards?
Yes, for sure. And this is interesting because it was a question I was asked during the Blueprints Pathway training program, if I had intended using this as a brain health marker. And whilst I had, I think it was really brought to my attention that that would be a very strong use. Because what we're aware of is when I get subjects, so when I said healthy control, let's talk about that.
Yeah.
All of us have different baselines. All of us have different pathologies. All of us have different
Yes.
things that we've done to our bodies over our lifetimes. And so
How
what's
healthy really are we? Yeah.
Question mark.
Yeah.
None of us know our baseline amyloid. None of us, depending on our risks and our lifestyles and our ApoE status. And
Yeah.
yeah, so being able to monitor the change for you
Yeah.
over your life is the most useful. So it really came to my awareness and has become a strong passion since training, honestly, because I had thought about it, but it was more focused on I want to be able to tell who has MCI, control an AD, but brain health monitoring is something we can't do. We can
Mm.
monitor our cholesterol, we can monitor our sugars. We should be able to also monitor our brain health over our lifetimes. I think that
Yeah.
would be amazing.
Yeah, that would be fantastic, and certainly kind of a promise of personalized medicine, something we've been talking about for decades now, but it will hopefully get there slowly. I mean, you see more and more of from different industries, right? So wearables, now to your point, we can track so much activity about how many steps did I take today, or on and on. And to your point, having a marker for brain health earlier in everyone's lifetime would be really interesting to be able to modulate behavior and adjust so many
Yeah.
opportunities
And
for prevention, not just pharmaceutical, but just lifestyle changes that patients could enact. So
yeah, and it's surprising, right, that we only have not surprising, but it's alarming or a big need that the companies who are designing therapeutics
yeah,
they need a tool like this because the only way they have to monitor if their drug is efficacious is to repeat PET imaging, which is exposing the patient
yeah,
to radioactivity or do lumbar punctures. I mean, who wants to have a big needle in your
yeah,
back every time you want to see if a drug is working? But then I also say to people who are wary of, well, we don't have a good, therapeutic yet. Well, we never will if we don't learn about the disease. So
yeah,
if we can learn more about the disease, and we have a big deficiency in understanding how it looks in, males versus females, there's a lot that we don't understand.
yeah,
A lot of our information needs to be broadened across the general community so that
yeah,
we understand the disease at large in the
yeah.
whole world, like and what it looks like in different populations.
Yeah. Yeah, no, absolutely. This would be, to your point, a great assistive tool for clinical, enabling clinical research in the pharma space. It just seems like there's so many applications as I speak to you. So this is fantastic. Really great. So you mentioned you're in the process of taking this, forward from a technology standpoint. It sounds like you're certainly working on improving performance of the test. But where do you see yourself and the team headed? Do you think you'll be more motivated to kind of t step into an entrepreneurial role and take this on outside of academia, or are you hoping to recruit some team members to help you bring this to market? What are your thoughts?
I think all of the above. So I was lucky enough to get an innovation award at CUN Shoots, and I have a fantastic follow-on team of mentors, which has been fabulous because it's kept the momentum going. From the Pathways program, I've stepped into having a team of mentors around me. So they ask me this question often, and my problem is I'm excited about everything and excited about the all of the applications that I can see. So as I've shared, I've developed this into cancer now. I'm working with people on autoimmune diseases such as lupus, multiple sclerosis. We have stiff person syndrome. We actually have the
Yeah.
neurologist who diagnosed Celine Dion is at CUN too, so I'm working with her.
Oh, wow.
Um,
Yeah.
so I'm excited about all of these. So how I funneled it, or how I am funneling that interest and excitement, is I am setting up what's called an exosome biomarker core so that I can have a team, like so I can have a person who will lead that core essentially, and then people who can obviously run the experiments, and I would train them all in, but then I can become aware of all the needs of all the diseases that are in need of early biomarkers. And we can help them work on that through that core whilst I, can dedicate at least some concentrated time to finishing developing this Alzheimer's drug. We, or Alzheimer's diagnostic, we are We just got asked to write a full proposal for Eisai, which is one of the anti-amyloid therapy companies. So they provide certain amounts of money to do research in general, but I've
Oh,
just asked them if I can get access to some of the plasmas from people who have been treated or not treated, or even just imaged, as they came in for treatment, because what I want to do is develop a cutoff so that at least we can get a feel for if you have this level of the biomarker, this means that you're probably more in an MCI range, or you're healthy, or you're closer to AD.
sure.
So
Yeah.
cutoffs are really important because we know our cutoffs again for our sugar levels and our
Yep.
cholesterols. So we don't have that for a biomarker. And so I'm
Yep.
wanting to develop that. So I really would love you can experience what the mentors experience with me because I want to do all of it. But I think a companion diagnostic would be really a very strong use of this biomarker. So we would be the kit or the test that
Mhm.
people who are giving anti-amyloid therapies or other Alzheimer's therapies to patients, we would be the measure to see if those have had beneficial or efficacious outcomes.
Yeah, exactly. In the clinical research domain. Yeah, absolutely. I see lots of application there and utility. Um, did you have a chance to apply to Blueprint, our funding for optimizer funding?
So I applied to the optimizer, I think it was the optimizer, but they deemed it human research, even though it's well, like for us when we're applying to NIH grants, because the subjects are de-identified and I'm just receiving plasmas. My understanding in any grants that we write is those are not considered human subjects anymore, but it was. Ming was helping me understand that they counted it as human research, which it isn't. So I think which is kind of complimentary. I th probably I'm next stage up from wherever I think I did optimizer and it's the next stage up, or incubator and I should be optimizer. I don't really
Yeah,
understand.
no, the incubator, sorry. Yeah, the our award is called
Yeah.
the Optimizer. It's the incubator program. So, you applied into pathways. We want to see more applicants like you coming from pathways then eventually to our incubator hub. So, that's why I was saying, did you apply? We had a solicitation over the late summer, I think August or September. Did you see that solicitation open? Okay.
Yeah,
Did
I
you
put in a
apply?
pre-proposal, but it was
Okay.
counted as human research, so it was
I
not
see.
considered because of that.
I
Um,
see.
but I do think there's a strong interest. So
Yeah.
if you guys can advise me on which form, maybe I'm further along because it is, I don't know. I would love advice because yes, we have gotten a score for an STTR on our first submission, so I'm excited about that. I've teamed up with the chair of biomedical engineering, who's an amazing grant writer. So I get excited about everything, and then I need someone to home me in and say, okay, these are the things that you want to see in a grant. So she's fantastic and has kind of taken me under her wing and is excited about this project. And we're
Yeah.
developing
Yeah.
ovarian cancer markers together. So, yes, so I would love advice on what to do differently. I think we were a little bit disappointed because I know the team really loved this project as
Yeah,
well.
no, I mean, I don't want to steer away from the conversation of what we're trying to do in terms of the video, but we can hopefully, edit all this out. But just as an aside, I think what maybe Ming is referring to is if some of these samples, specimen samples that you're collecting had been already collected as a data bank, if you will, and if it could be then IRB exempt, essentially that would help. I think it's the active recruiting data samples and building your data bank. That's the piece that he's speaking to is not allowable in the incubator hubs. We won't support things like that, but we do have projects that come in with existing data sets, for instance, and where we allow research to or development, excuse me, to continue because that would be an IRB exempt, for instance.
Yeah, because we have existing cohorts. So in the pre-proposal,
Yeah,
I shared that we had existing cohorts that I was going to use, but
yeah,
unfortunately, it was still counted as human research, even
too
though I
bad.
don't think it should have been. But
No,
you
exactly.
can
I
dig
think.
into the pre-proposal and maybe advise. That would be.
Yeah,
I did.
yeah. I think maybe on the next cycle we can try to set up something with Ming and my. I'm happy to hop on with you as well, and maybe we could just brainstorm a little bit about how to position this because it does really sound right up our incubator program alley, meaning we should be looking at innovations such as yours. So, but thanks for that. So maybe
You're welcome. Yeah,
we'll
I'm excited for advice and
yeah,
guidance.
yeah.
Yeah.
So we'll try to veer off from that tangent and come get back on track here. So yeah, I think you mentioned a few things about what you're up to with your team. The core imaging core, that's really exciting. But as far as you said mentioned SETR, so can I ask you a little bit about the test eventually? Will this be essentially a blood-based sample? So you could go into outpatient setting. This is just a simple blood draw. You're running this off of plasma, right?
Correct, correct.
Yeah.
And so I have a couple of thoughts on potentially looking at maybe putting blood drops onto paper, but at the
Mhm.
moment it would be a blood draw. I actually even thought about, having people send in urine, but neurologists weren't as excited about that. They kept telling me that that's a long way from the brain. So they're still happy with the blood draw. They like the idea of being in control of the blood being drawn after they've met with the patient and so the controlled blood draw, but it would, as you alluded to, be something that a primary care physician could do.
Yeah.
And then it would either go to, a company like Quest Diagnostics or something similar where they run the test,
Yeah.
or it comes to us if we were to set up a little company to run tests for people,
Yeah.
um, or it's something within the same home as the pharmaceutical company and they run it in-house
Right.
alongside their
Right.
treatment.
Yeah,
So
that's kind of where I was heading with my line of questioning. So interesting. So obviously, if you own the IP, that's great. But in terms of eventual commercialization, is this sort of test something that you could pass on to Quest and have them develop the proper assay? There's nothing too proprietary that you would need to produce. Um, per se by yourself. This is all existing assays and reagents that a Quest has access to.
So there would be all existing components that I was asking about this during training actually as well. So how do you, I think it was Andrew that I worked with quite closely thinking about this. If we take someone's beads and someone's, this component, how do we put these together and stay within regulations? So I think it
Mhm.
is allowed. I think you obviously have to talk to each company and tell them what you're thinking of using it for.
Right.
So I would need some help understanding that process, but
Yeah.
I could very much set up a protocol that's
Yeah.
very easy to follow. And I have shared this with most recently a new undergrad who started working with me, and she can do it all the way through. It's not a problem. And you use such tiny amounts of the blood or the plasmas that you can do millions of tests, like just thousands of tests. So if we think about a 110 ml blood draw giving about 5.5 mils of plasma, of that, I probably end up with about 6 mils of exosomes. That's back of the envelope in my head. And then I only need 50 microliters to do the test itself. So lots and lots of tests from one blood draw. So
Yeah.
that the only limiter is understanding the biomarkers that are good diagnostics. And so
Correct.
that's why I want to expand across different diseases, which
Yes.
core would allow us to do.
Right.
But um,
Right.
yeah,
Right.
so
But and so that test, that's the piece that is the commercial opportunity, correct? Meaning that would be proprietary to you and your core, and as part of the license, you could send all of these out to any diagnostic like a Quest or et cetera, right? LabCorp, anyone who's running these diagnostics. Okay, great.
Absolutely.
Yeah,
Absolutely.
no, it's a great model. And did you mean Andrew DeMeo? Is that who
Yes.
you worked with?
Yes.
Oh, yeah,
Amazing.
Andrew's
Just amazing.
yeah, Andrew's awesome. Uh,
Hmm.
Andrew's a great mentor. So that's great. I'm glad you got some one-on-one time with him. So that's a perfect segue into, and you've been mentioning it all throughout the interview about how Blueprint has been able to help you think through this innovation, thinking through how to bring it to market. Um, what do you think are some of the doors maybe or avenues that we opened up for you as you interacted with our program?
I think it was really getting us focused. Just, we all came in with this excitement for all of us had excitement about our projects, but really starting to home us into what is the most exciting part of your technology to you currently, because you have to start small and then work out to bigger. So that's why I always correct myself when I say I'm interested in lots of things and lots of diseases, is just that reminder that if we can successfully roll this out as a test for Alzheimer's, then that builds confidence in your technology. It builds
Mhm.
confidence in you as a researcher. I think they were also incredibly good at helping us pitch our ideas, because as researchers, we tend to tell long stories over an hour in a presentation, and we had to learn how to really capture our audience in five
Mhm.
minutes. Because
Yeah,
if you don't, they're gone, right? And that has lots of translatable skills associated with it. Writing our STTR, I have no doubt that that helped because I knew what we needed to bring the audience's eye to when we wrote it.
great.
Um, the same, I'm about to help someone as the student who started working with me we got invited to give a talk at a conference next week, and she's working on blood-brain barrier, and I'm helping her pull a pitch together. I said, I'm going to help you with all of the skills that I've learned, catch the audience. And so that is a skill that I think is, and for researchers, your tendency, I still laugh. I think it was Andrew again or Ming that, we want to share every piece of data, even the data we just got this morning that we don't understand yet, but we're going to share it anyway. And they were like, really not useful. That might be exciting to you, but you can't even explain it. You don't even know what it means yourself yet. And so just reminding myself, it's like you sell, when you're pitching, you are literally selling like the excitement that you have for this project to others.
Yeah,
What is the most exciting part of it for you?
yeah, yeah.
Uh,
No, absolutely.
so that was huge. I think that was hugely helpful. And I have seen it. I mean, I have seen that change how I speak with people about the project, how I think about what is important about what I'm trying to do. We got donor money because of, being able to share with them what our mission was. So I think it really is going to have downstream benefits as well, just from being able to afford to do more of the research, which is the goal, obviously.
Right. That's fantastic. That's exciting, and I'm glad to hear that a donor was moved to help support this critical work of yours. That's great. That's exciting, and there's lots of ways to, as you just said, fund this work. So it looks like you're looking at all avenues, which is fantastic. Federal, non-dilutive funding, donors, if that's possible. Um, so as you look out, where would you like to see this in the next few years yourself?
I think what's really exciting right now is that, so I wrote a pre-proposal for this ESI. I think
Hmm.
if they would allow us to have access to PET imaged individuals, even if they won't give us people who are being treated, because that makes sense. They obviously want to have more control over how those samples are handled. They're very precious. Being able to come up with some cutoffs so that we could start to think about ranges of what a healthy brain looks like in terms of this biomarker. Also developing other markers that give us more confidence. So I'm going to do metabolomics and proteomics of these exosomes that are taken from people who have disease and not, because that will help us start to think about designing new therapeutics because I'm a pharmacologist by training. So I want to understand what is changing as disease develops. And
Yeah,
so then that provides more insights into what other targets we should be thinking about to
right.
intercede in the onset of this disease in the first place.
Yeah,
Hopefully, we'll have a world someday that never has, people
right.
never have to experience Alzheimer's.
Absolutely. Well, I for one am ready. So when you're available, I'm ready to give my blood sample, have it screened and evaluated. So uh, hopefully you're close to Maryland somewhere. If not, I can find my way to you.
I
Uh,
like TC. I can fly out for blood.
so well, that's great. And we've touched upon this a lot, tangentially, but just to kind of speak about the industry and where you see diagnostics going, I'm sure you think about this a lot. Can you maybe speak to what you kind of see unfolding in the next few years, 5, 10 years for diagnostics overall?
Yeah, I think we are very aware that we need to move in a direction of blood-based biomarkers for all disease. And
Mm,
that's because we need to learn about the community of large for all diseases. But we also, a lot of tests that are otherwise prescribed, I guess is the right word, by a physician are not always going to be approved by insurance companies. So if you're someone without insurance, we're starting to introduce disparity in healthcare, which I don't like. So I want a test that is available to everyone. I grew up the eldest of six with a working class family, and my mom and dad didn't even finish high school. And so when people ask me how I ended up doing a PhD and in this field, I think it was stubbornness, excitement. I remember my mom being asked by a neighbor, well, why is she not just going to take over her aunt's little corner shop? And her answer, to this day, I can remember it. She said, and you've met Christina, right? And so I always had this fire for science. I watched this show called Tomorrow's World about bacteria from the age of 8. I can remember knowing about bacteria and germs and science and knowing that that's what I wanted to do. It had to be a part of my life going forward in some format. Um, and I've always loved people. So I trained as an AEMT. I was actually quite good at getting blood out of people. It's really hard to do science and AEMT work together, I found. It's hard to do 12-hour shifts on both. And so
mhm.
I just love like meeting the person that needs the test. So I think the pivot for me, I actually was shadowing one of the neurologists who I was talking to about this test. And he had this 50-something-year-old. She was super healthy, super athletic, but had had some quite big memory lapses the week before. And when she left, he said to me, Chris, that's who I need the test for. Because no matter what I say to her, because I have not objectively measured something that tells her that her brain is okay, she is going to worry about this now forever. Like she knows that this was not what should have happened. She shouldn't have forgotten where her car was or there were big memory lapses. Um, and so I think that was really the impetus. I went back to the bench with, I think, more gusto after seeing that person because I
Mm,
had a better visual of who it was that I would be helping. And that's often healthy individuals who, like yourself, or, have someone with a disease, are worried about, my risk because this is in my family. I think I like the idea of educating people just in general. So I've helped with the
yeah.
Alzheimer's Association local chapter just to remind people, 65 and above, if someone develops Alzheimer's, we think of that as more sporadic. If somebody is under that age, then we think of genetics a little bit more. But just really educating people as to what we can test, what we can't test, what we can do, and adding this as a test to give people peace of mind would be amazing.
Yeah. Yeah, absolutely. I think if we can continue to incorporate these kinds of diagnostics earlier and earlier, as you point out, I love the analogy of cholesterol. We start testing for that so early, generally in populations. So just moving that needle into brain health would be fantastic and a huge win for so many families, that have to deal with the downstream consequences, as we all know all too well. So,
So, my name's John Simmons, and I'm the CEO of Cool Spine. I've been in medical devices since my earliest days after college, as a matter of fact. I started, believe it or not, with Procter& and Gamble selling soap. Um, and Procter& and Gamble then went into disposable drapes and gowns, and they asked me to join that group. So then I went into hospitals and got trained and worked in there, which was a big transition, but really which started my love of medical devices and hospital care and doing stuff for patients. So then I left P&G, went to business school, and then um, joined Becton Dickinson for many years in the diabetes area, both in the United States and Europe. And I was the development guy for diabetes in Europe, Africa, and Middle East for about 10 years, and then switched over to their mainstream medical devices and came back to the United States with BD, worked in pre-fillable syringes and drug delivery mechanisms, which was very interesting. And then left BD, went to J&J and started in a startup of J&J's, which is a sterilization for hydrogen peroxide gas plasma. And we were applying that for medical devices such as pacemakers and things like that. And that was working for J&J was fantastic. I then left J&J, went to Dexcom, which I'm sure many people know about, which is the glucose sensor. I was in the early A and B rounds at Dexcom and down in Southern California, and that was very exciting. I then left California. My kids were getting to that stage where I said, I got to get them out of California. But we came back to the East Coast and worked for another startup in in containers and pre-fillable vaccine containers, which we sold to Glaxo, which was a successful exit, and then went to looking for the next proverbial gig. I went over to Yale and to their Office of Cooperative Research, and they said, we just don't do much in devices, but we know this guy. And they said, he's got an interesting idea. And it was Dr. John Eleftheriades, who is a worldwide thought leader in diseases of the aorta. And we met and talked, and he had the idea for Cool Spine, which I'll want me to just transition into
yeah,
Cool
absolutely. It's
Spine now?
perfect segue into tell me about the
Okay.
company's mission and how it was founded.
So, Cool Spine's mission is to provide neuroprotection to the spinal cord or to the brain by inducing localized hypothermia. And we do this by cooling the cerebral spinal fluid that circulates either in the brain or the spinal cord. So we have two products, an intrathecal cooling catheter, which is part of the Blueprint MedTech program, and the point of it is it's being used during thoracic aneurysm repair. And this is where Dr. John Eleftheriades was an expert. And what he saw, and what everybody sees, is that the rate of paraplegia during thoracic aneurysm repair is horrific, and it remains horrific. It's from 6% at a really good institution to 15% in certain types of procedures, depending on the complexity of the repair. And it's in both open and endovascular repair. So his idea was to induce localized hypothermia during the procedure. And we do this, it's a catheter-based system, and we do this by cooling the CSF, the cerebral spinal fluid, which which just surrounds the spinal cord. And by cooling the CSF, we drop the temperature of the spinal cord to levels known to be neuroprotective, which are 32 to 35 degrees C. And we do this locally, and the key benefit is we maintain systemic normothermia, which avoids the complications of systemic hypothermia.
Right. Oh, wonderful. So when you met John, uh, where was the stage of this entire innovation and the platform? I mean, are
Okay.
you start from
So
ground zero, or was there
it was pretty
some
close to
engineers?
ground zero. He had done one animal study in which he developed a 2 lumen catheter in which he circulated the cold saline, and he was able to demonstrate in sheep the ability to cool the spinal cord to neuroprotective levels. So that was the first, proof of principle. And then we joined together with another neurosurgeon, Rohit Khanna from Daytona Beach, Florida, and he's a prolific inventor. And we started Cool Spine. So that was a while ago, and we've got a few NSF grants and proceeded to develop two products, the spinal cooling catheter and the brain cooling catheter, and we developed them and tested them in large animals. We continued the focus on large animal testing. We picked sheep because of the similarity of a sheep spine to a human spine. It's a very good model. And then we obviously went to brain because we're cooling the CSF. The CSF is either in the spinal cord or the brain. It's all connected. But if we could cool the brain, we could address the horrific brain injuries of ischemic injury. And remember, ischemic injury is what we're trying to address,
Yeah.
where the blood flow to these organs is reduced either through like surgical procedures where they clamp the aortas and things like that. And when the cerebral flow is reduced, oxygen is reduced,
Yep.
and that starts the process of cellular death. So when there's not enough blood flow, the neuronal uh, uh, problems occur, and that's how you get ischemic injury. So by cooling them, we're delaying the onset, if you will, of ischemic injury with hypothermia.
Right. Absolutely. Great. No, that's wonderful. And have you had some development team members join and help with the engineering effort, or are you outsourcing all of that? How's your
Yeah, that's a great question. We have been outsourcing. We're not we're not well funded. Okay, um, and it's an interesting story. If I might diverge a little bit, the VCs put a ton of money into hypothermia in the early 2000s, and they funded three companies in a large way, 100 million apiece, and they all went after brain injury, stroke, which is, you probably understand very well, is very challenging. It's just, and so they all failed, and more or less, and they all got black boxed by the FDA. So we've always focused on going with spinal cord injury first, with thoracic aneurysm, obviously, so it's prophylactically employed and at a much lower risk. But getting back to your question, what we've done is we've outsourced our support work through a variety of several different medical device companies, both a Microspect for catheters and Sage Medical will be working with us going forward. And that's what we've done so
got
far.
it?
We've
Great.
outsourced stuff.
So it sounds like strategically there was an opportunity you saw, as you just said, in terms of medical applications. People were really focused on cortical sort of brain injuries and pathologies associated with addressing really your primary cortex kind of injury. Whereas you focused and your team focused on the spinal cord. And were you the first group to really uh, bring a cooling based sort of innovation into that space?
So I'm gonna just to be clear, the people brought cooling to the brain, okay, but they did it by blood cooling, systemic cooling. And systemic cooling starts off a process. Uh, the body reacts to the total body temperature, and you have adverse events. And that really stopped systemic cooling to be used. It really stopped them from having any success in the brain because of the adverse events, which is tachycardia, coagulopathy, pneumonia. You imagine, pneumonia is huge. And
Yeah.
what we do differently is we approach it local cooling. So by cooling the CSF, we're only cooling the targeted tissues or organs
Yeah.
and maintaining systemic. So that was the big, huge difference with
I see.
us
Yes.
in the approach. And interestingly, at Mass General back in the 90s, two great thoracic surgeons did something similar. They infused saline into the spinal canal to cool the spinal cord and had very good results. Unfortunately, it was very difficult to do. Imagine infusing saline. Someone said it was like working in a swimming pool. And while it had excellent results, it was never able to be actuated on a broad level. It was too difficult. So what we have done, the innovation that we came up with, was to cool the CSF with a tri-lumen catheter. And the saline stays within the catheter. It circulates in a closed loop.
Yeah.
It's in the spinal canal. It cools the CSF, which in turn cools the spinal cord or the brain. And so by not putting anything into the body, it's a catheter that's very simple, it's an intrathecal catheter, much like a spinal drain. And as a matter of fact, the other innovation of our product is we incorporate a spinal drain into our system. So there's three lumens, two for the cooling, one for drain. So the procedure for draining the CSF is a standard procedure to reduce ICP. So our system can be employed without an incremental intervention into the patient because you're going to have it done anyway. You're going to have a drain put in to manage ICP. So we
Got it.
can do both cooling and ICP management in one intervention in one catheter.
Awesome. Wow. So that leads me to really then ask more about your target beachhead market, and because it seems like this is something that's applicable to a broader neurocritical care population. But where are you starting?
So we're starting with repair of thoracic aneurysms.
Mhm.
And the one of the reasons, obviously, is Dr. John Eleftheriades. But the interesting thing about thoracic aneurysm repair is it was discovered in the 60s by DeBakey, Cooley, and Crawford, and it was a huge pioneering aspect of aneurysm repair. But the rate of paraplegia was still huge back then because if you think about it, you're clamping the aorta and reducing blood supply. That hasn't changed, even with endovascular repair. So the need of neuroprotection during thoracic aneurysm repair is immense, and everybody recognizes it. I mean, one of our partners had an international symposium on it 2 years ago at Ohio State University, and I can talk about that a little bit later. But that's our beachhead market because of the huge unmet need and the recognition by all practitioners of aneurysm repair that they need something desperately. Honestly, I can say that when we talk about this innovation to a physician, I've never met one who hasn't said, I want to be on the clinical trials. I mean, that's how serious this unmet need is. The other aspect of it is brain. We decided not to go with brain initially just because brain is so much harder, and
Yeah.
rightfully so.
Right, right, absolutely. And so, if in this patient population, in that surgical context, I'm don't want to make any assumptions, but is it safe to say that this seems really easy from a workflow standpoint to deploy, as you've already said? Can you talk a little bit about that and how John, if any, has been able to comment on where he sees this fitting in procedurally, and will it extend any procedure time? Those kind of aspects.
Yeah. So yeah, from a procedural point of view, it's almost seamless. You're putting in the anesthesia for thoracic aneurysm repair. It will, the team makes a decision, but the anesthesiologist will insert the intrathecal catheter just like he does a spinal drain. Okay, so there's going to be, and they'll use fluoroscopy more and more, and it'll be safely implanted temporarily during the procedure. Now, the interesting part, so from a patient and physician and workflow, there's no change. Okay, so which is great. And
Yeah.
as I said, with one intervention, you now have both cooling and draining. The other aspect of this, which I think is critical, is our system. We're flowing saline in a closed loop, so it's like an IV pump. Okay,
Yeah.
so it travels with the patient. Okay, so you can, during the procedure, obviously you'd be cooling to induce hypothermia, but oftentimes there's delayed ischemic injury, which is equally as problematic. So this could follow the patient back to the ICU recovery room, and if they see incidents of delayed ischemia, uh, they could restart the cooling to address it. This is particularly critical for brain cooling because, like for an example, in subarachnoid hemorrhage, the incidence of delayed ischemic injury is as bad as the acute ischemic injury during, that happened initially. So
Yeah.
this is a real benefit of, this system traveling with the patient uh, and being used when needed.
Right.
And obviously be in the hospital though.
Right, right, absolutely. And that speaks to the neurocritical care application,
Exactly.
which I was hinting at. So
Yeah.
it seems that there's broader application, different populations as you expand beyond your beachhead eventually. Um, that's really great. So, in terms of your next features or partnerships that you need to kind of make all of this happen, both inside of Blueprint and then beyond. Well, what are some of the areas that you're looking at as a team?
So the first So we have two good partnerships in this particular program, which we're very excited about. One is kind of a clinical partnership, and one's a development partnership. The clinical partnership is with Ohio State University and Dr. Hamdi El Said Awad, who is like a world-class thoracic anesthesiologist. So we're going to be able to do all of our animal studies out at OSU with he and his team and which is recognized as a world-class team. So we're gonna learn a lot from that. The second partnership is we're working with Dr. Alex Slocum of MIT, who runs the medical design class at MIT. And we're going to be partnering with him to help us develop the next iteration of what we call the controller, the pump and the controlling of the cooling of the saline and stuff, the stuff that's on the IV pole, so to speak. And working with those guys at MIT is fantastic because first off, you tell them, oh, we think you should do this. And they say, well, wait a second, we want to do this too. We have another idea. We have another idea. And it's like, I'm like, uh-oh, project creep. But it's like, whoa, I'd be crazy not to listen to these guys. So, of course, they're all looking at real-time data management, which I think, will be an outcome, and which we worked on in our seedling program to address that. So we anticipate that in this next phase of the blueprint, the real-time management of data coming from the patient in terms of cooling and things of that nature and duration and depth of cooling is going to come out of the work we're going to do at MIT. So we're very excited about that. And then future, uh, you know, there's two other aspects that AI, obviously, we all have to think about AI. And I think AI for us is going to be more after a lot of experience, in terms of getting patient data, and which we're starting to do that with Dr. LF Teriades. And specifically, what that'll give us is things like this will maybe tell us better in advance when ischemia is starting to happen because it's not easy to figure out during a procedure, right? You know that maybe there's reduced blood flow, but you don't know what's going on. So the I think, may be able to give us greater warning, so to speak, that, hey, maybe we need to go a little bit deeper or longer in the cooling. Um, the final iteration is for perhaps military applications, which both traumatic brain injury and spinal cord injury from concussions, from shock or trauma injuries is huge, and there's a need for that, and that would be after our commercial, if you will, or patient clinical uh, uh, products have been demonstrated.
Right, absolutely. And you just hinted on some aspects of Blueprint, but can you speak to how Blueprint has helped shape the journey for Coolspine in the last year or a few, as you mentioned, in Seedling and even in the upcoming year? Well, what sort of impact?
So we were introduced to Blueprint by NINDS, and we had made a proposal to them, and one of their guys, I won't mention names now, but he said, I mean, you're sure you know who it is. But anyway, they said, John, you should really look at this. This is a really good idea for you. So we did that, and getting into the ecosystem of Blueprint was fabulous because we had uh, innovators in neurotech who, were passionate and both from an academic level from Hopkins, uh, who were like all over us asking us great questions. We had people um, who had been in the business who say, hey, here's some great ideas. We had the NIH people who would always come in and, you know, give you some thoughts. So the whole, and people who had been there and done that. So that has been the real benefit of Blueprint is that people from a diverse uh, experiences, academic, clinical, regulatory, and so on and so forth, and management have helped us focus our approach and made it tighter. And the seedling program in particular enabled us to, as I mentioned earlier, go after the real-time data management, which they recognized as a weakness in our early stage. So that's what we did. They also helped us, uh, focus on the regulatory component and just make it tighter because we've always worked very closely with FDA. We've done a lot of pre-subs and Blueprint said, keep on with the pre-subs and just tighten it up and let's make sure that we have a regulatory plan that we can take it to FDA and they go, yep, that makes sense, and maybe get a positive designation, which, I obviously will be going for a certain type of designation in the future. Um, and so that's where the seedling program really in a compact period of time, got us to focus on the right things and, make a better proposal.
Great. Excellent. Yeah, I'm excited to have you guys now starting the optimizer
Yeah,
period and get really rolling. And you
we
mentioned,
can't wait, honestly.
yeah,
We're
and you
like,
mentioned
oh, let's
already,
go.
I think it was Sage and a few other partners that you've used along the way. In the optimizer period, will you be working with Sage and a few others
Yes,
on your work
yes.
plans?
Sage, Evera, which was a great regulatory group that we found that you guys approved, we used, and we're going to continue with them. So yeah, so we're continuing with all of our seedling people, and then adding on,
Excuse me.
OSU and MIT in particular.
Got it.
And going back to some old great standards that we've had, like microspec on the catheter extrusion stuff.
Yeah, absolutely. And so you did mention FDA, and I'm glad we were able to, kind of extend your experience with working with FDA. Can just speak a little bit about where you see the direction of this going? Will this be a 510(k) or is it de novo? What is some of the feedback?
It'll definitely be de novo. Okay.
Okay.
There's, we, as I said, we've had uh, three pre-subs with FDA, both on brain and spine, and we've worked through this with them, and it's clear that the cooling and the draining is a de novo. There's no
Yeah.
predicate device.
Right.
There's no way that anything is at 510K in that kind of area, and that's
Right.
that's fine. Uh,
Well, that
we
just
think
speaks to
it,
the innovation,
yeah,
actually.
it could
Yeah.
be breakthrough. I mean,
Yeah.
it's too early to, obviously you got to give them the data before you do that. But that would be nice.
But
But our so far our work with FDA has always been great. I mean, in the sense that, we go there, we tell them what we're doing, we ask them, you ask the right you gotta always ask the right questions, right? You
Yes.
just can't say, tell me what you think.
Sure.
Um,
Sure.
but you read them,
Right.
and um, they're like, okay,
Well,
like, oh,
that's
that's good.
fantastic.
You know,
That's
that's a
fantastic from a company standpoint. That's going to help from I'm sure I don't have to explain to you a competitive advantage if it is de novo. It's a
yeah.
novel technology you're bringing to market, so hopefully will allow you a lot of commercial success as well as you get into that phase of your life cycle. What I'd like to hear a little bit about is, maybe coming back to the field of cooling. What do you see happening competitively out there? Are you still, fairly ahead of the curve on this, or what do you think is going to unfold over the next few years in this space?
So it's a good question. So I gotta say, cooling per se took a punch in the mouth in the early 2000s because of the big failures of the VCs
Yeah,
on their blood cooling products.
yeah,
And so as a result, VCs are like, well, no, it's no longer in 2000 it was the shiny new object. And then they got punched in the mouth. They sold for pennies on the dollar. And now there's a bunch of systemic cooling products out there, really fever management type of stuff, which people don't, because of the adverse events, the adoption is not spectacular. So I think our patent portfolio protects us greatly with our approach. We've got a very good patent portfolio. And I think based on, the reaction both of thoracic surgeons and neurosurgeons on the concept of, hey, we're cooling the CSF to reduce the temperature of the spinal cord of the brain for ischemic injury. They're like, oh, yeah, that makes total sense.
yeah,
When we were at Boston last year at the annual meeting, I was talking with a very, spectacular, well-known neurosurgeon, and our whole poster presentation was about spinal cord. And he said, well, what about brain? I said, yeah, absolutely. And I started to talk to him, and he said, what's your first application? And we had thought about doing other like TBI, and it's just too damn complicated. Like TBI's got so many different aspects of the disease. I said, well, we talked with FDA and they really said, go after subarachnoid hemorrhage. And he goes, exactly, exactly. That's what you should do. And I'm saying that he goes, now you tell me when you're ready, and we're like, oh my God, he couldn't have a nicer conversation.
yeah.
And interestingly, one of the persons from NIH, one of the core people from NIH was listening in. And I turned to that person and I said, did you hear that? She goes, yeah, I heard that. So it's like, by the way, the meeting in Boston was fantastic. It
Great.
really was. I mean, it really exceeded expectations with the Spalding stuff and
Yeah,
everything like that. It was just, it was really motivating. So that
good.
was great.
Excellent.
Yeah.
Excellent. And we're looking forward to having you come out to DC in a few weeks.
Can't wait.
Yeah,
Oh,
absolutely.
hopefully, hopefully it's open, right?
Yeah,
Okay.
yeah, exactly.
So,
Well, at least it will move. It will definitely be
no, I
moving
know.
forward,
That's
but
a
it's
bad
just
joke.
whether or not NIH will be able to attend or not. But yes, the meeting will continue. But yeah, no, John, I mean, I asked a lot of the questions. I feel like we canvassed pretty much the whole area that I wanted to explore in the interview. Do you think there's anything that I didn't really touch upon in terms of what Cool Spine is up to that you want to mention?
I don't think so.
Yeah,
I think we covered everything that
yeah,
is on my list. We're I think actually one thing that's fascinating about this is on our first application of disease of thoracic aneurysm repair and the rate of paraplegia. What I don't think I never appreciated is when a patient goes in for a repair of an aneurysm, a thoracic aneurysm, and comes out paralyzed. That's clearly, we all understand it's a devastating complication for the patient, their family, and everything like that. And affecting life issues and expectancy. What it's also a incredible challenge for is the surgical team, the doctors and the nurses. When this happens, it's really a post-traumatic stress-inducing event. These nurses and doctors and anesthesiologists are greatly affected when this iatrogenic injury takes place. And this is something in neurotech, in neurology, I think, which is much probably more prevalent and where we can begin to look at much greater effect with our advances in neurotech, these iatrogenic injuries that are happening to patients during surgery. And, ours is a very basic neurological deficit, right? I mean, ischemic injury, and there's others. So I think Blueprint's focus on neurotech is going to not only allow us to go after the really exciting, the fun, the stuff that's really out there, but also going back to basic clinical needs that we haven't quite addressed yet.
Yeah.
And so if you think about neurotech, and all the advances that are coming up, or potentially, you can't have neurotech without saving neurons, right? So that's kind of the level where we're at, and I think Blueprint will perhaps give more focus to saving neurons, so then we can repair neurons. So that's kind of my like you get excited about that.
Got it. Excellent. No, that's great. That's wonderful. That's a great way to view the field and what we're all working towards.
Yep.
So very well said.
I'm Rodolfo Finocchi. I'm one of the co-founders and CEO of SafeSonics. I have a background in biomedical engineering and robotics, as well as an MBA, and have been working in medical device startups as well as large corporations throughout my career. So I'm excited to be part of this and talk a little bit about SafeSonics.
Yep. Nice to meet you. Like my name is Jun Kang. Like I'm an assistant professor at School of Medicine at Johns Hopkins University. And my background is electronic engineering. And then I'm the one who was really bored with all the like ultrasound imaging instrumentation happening. And then like I was like looking for the new form factor. And then this project is really making my dream come true. So really nice to meet you.
Right. Absolutely. No, we're glad you're excited to present and talk about your work today. So, who would like to get us started? So, first off, with just about the company's mission and how it was founded, maybe a little bit of the origin story of how this all came together. Initially, maybe out of the research context, and then we'll get into maybe company formation as well.
Sure, I'll take it.
Okay.
June was the one was involved in most of it. I came a little bit later, but just to give you a high level, yeah, the company's mission, it's to use automation and advances in medical imaging to monitor and protect babies' brains when they're at their most vulnerable. And so in 2016 is when the journey started at Johns Hopkins. Members of our clinical and our technical teams came together and identified a big gap in the technology used to monitor brain health during this period when our brains are the most vulnerable. Labor, which is a very stressful and harsh environment on a neonate or baby's brain, um, during that time, the technology used to currently monitor the brain health has its limitations. And so the team identified significant clinical gap, an opportunity to improve and further develop the technology, um, and provide really more information and better information to physicians. So just to give you a little background about what the standard today is, it's electronic fetal heart monitoring. It's the current standard of care for monitoring fetal heart and brain health during labor. But retrospective studies have shown that it really has a very high false positive rate of over 99% detecting particularly brain injuries. Um, and this is something that the technology was actually developed to do. So clearly it's failing in delivering the promise. Um, the The rate of C-sections has skyrocketed since the use of this technology back in the 60s. It's become ubiquitous, being used in about 85% of labors today. But at the same time, the rate of neurological disorders such as cerebral palsy has stayed relatively unchanged for the last 50 or 60 years. So our mission originated from this, knowing this and the curiosity of our team, which of course includes experts in medical imaging and instrumentation, neurophysiology, obstetrics, robotics, and commercialization. And we conceived of a tool that harnesses the potential capabilities of photoacoustics and ultrasound imaging. Uh, we have compelling preclinical evidence to measure fetal brain oxygen saturation, and now we are further translating our idea through our company, Safe Sonics, and hoping to solve key unmet needs, which we identified during our customer discovery conversations with physicians, patients, and hospital administrators. And now the technology is a wireless wearable mechatronic device that monitors the fetal brain during the 6 to 10 hours of labor without the need for manual scanning by a sonographer.
Oh, fantastic. That's a great story, and to dive into that maybe a little bit more, what really ultimately inspired this direction? I mean, were there other ideas being considered with this technology when you were in early days of kind of forming what applications you could address, or was always the goal?
Yep. So I will take this question. So yeah, the brain is a mysterious and fascinating organ for us every time. And then it is sensitive and dynamic in nature and designed to adapt to surrounding environments and any external stimulations. It has been the way we learn, grow, and interact with the world from the first day of our life. The idea for our fetal brain monitoring device came like a destiny for us. Our team had developed the foundational neuroimaging technology for science using ultrasound and photoacoustics. And from